Okay, so I did post it, right? That's the ones that were on the exam, right? But nobody really cares about that because I'm basically going to give you a different one for the final. But this is what I just handed out in case you lost it, but I would highly suggest bringing it back with you because there are going to be a couple questions where you're going to need to use this code that's at the bottom, and we're going to go over that today. I'm kind of waiting for Michelle to get her butt back in here. So I'm going to talk about Barbara McClintock first, and then I'll come back to that. So have that handy. If you tucked it away, take it back out because you're going to need it for a little activity we're going to do. Okay, so that's biotech. So the other thing you'll notice that I did post for you guys, you'll notice I posted like an insane amount of stuff under this module. I posted all the outlines, right? All the animation PowerPoints, all the lecture PowerPoints, right, that I hadn't posted yet. Why are these not linking? Oh, this was just a list, right? This is just the list. That was just an abbreviated form of the list. There's chapter 12 right there. 12, 13, 15, and remember 19, and that's partly why I put this list. We're only covering section 3, transposition, and the experiment, 19A. So that's Barbara McClintock's. So I'm going to open up that lecture and scroll down to that section. Where is it? There we go. So this was pretty amazing stuff in her time. Let's see, transposition. So this involves the integration of small pieces of DNA into the chromosome. And sometimes these are also referred to as jumping genes because they did just that. They move around in the chromosome. So they're mobile. They're referred to as transposable elements, or sometimes, as I said, jumping genes. And they were discovered, and think about this time, because remember around the 1950s was when Watson and Crick actually figured out the structure of DNA. So, I mean, we're not even to like DNA sequencing or anything like that at this point, right? And so she was working, and a lot of the pictures that you see of her are working under the microscope. And so she discovered this phenomenon of these transposable elements. And she was studying maize corn. I'm trying to remember if they have a picture in here for that. Oh, just one little kernel. Oh, here we go. It's not the best picture, though. Oh, I meant to steal a better picture. So the kernels themselves, right, are each little new beginnings for a new plant, right? They're basically the seeds, you could say. And for some of them, she noticed this phenomenon where the colors were disrupted, and genetically, these were from the same parents. How is this happening? So at the level of the chromosome, she noticed things. So she literally, and they don't show pictures of it here in your book. I wish they did. I could send you guys a link to another website that I've used to read up on her. I should have made one of you guys shut the door on your way in. You got it, Tyler? Thanks. What are y'all doing today to me? I was all messed up. Okay. Don't close it all the way, Tyler, but close it because there were some people talking. Thanks. She can get back in. All right. So she was actually looking at the chromosomes under the microscope. And she had developed techniques, and I mean, she was an insane microscopist. I mean, she was just like, you know how some people are just talented at things? She was talented under the microscope. And so she was able to see these deformities to the chromosomes and notice this breakage that happened a lot to this chromosome number 9. And this is one of the chromosomes that affected the color of the kernel of corn. And so what was happening is that sections were moving, right? You had these breakages, and what would happen is when that segment inserted back in, it would sometimes disrupt the part that determined the color. So in this case of this kernel, right, it's disrupted where it's clear, and the red patches, it's no longer disrupted. And so there was this segment, right, this transposable element, or called a transposon, that was moving from one part of the chromosome to another, and when it moved, they tend to move into particular segments where there are sequences, what they call flanking sequences, where it will insert into. As you guys prepare for your test, I definitely want you to read over the rest of this section, right, and learn about the different details of the mechanisms on how this works. So this is just an overview of it. But then it would move again, right? So in the beginning, when the kernel was formed, it wasn't producing any color. But then that transposable element left that segment of the chromosome and went someplace else, and therefore the gene for color was no longer disrupted, and so you get these red patches here in the kernel. And I was kind of bummed because in your book they zoom in. Actually, I have access to the book. Why am I not doing that? Duh. So one great thing about the book being online, I can just click on it. So chapter 19. Yep, and see, that's where I left off in that chapter. Please tell me that picture was in my mind someplace else. Okay. So there's another really good website. So we'll just search Barbara and I'll put it on here. I always spell her first name wrong. Yep, this is it. So this has the pictures under the microscope. So I'm going to put this link for you guys here. It's a really cool link. Modules. Spell her name wrong. I put too many L's. Okay. Turn it on. Okay. So, so she was noticing, like, literally, she's looking at chromosomes under the microscope, y'all, and noticing these phenomena that were happening. And then, of course, she did just like Mendel. She did crosses, right? So there was a picture. Maybe it's in the problems. Yeah, there's like a little interactive here. It's cool. Ask you questions. Ah, here we go. I think this is the one that has it. Or not? Oh, you have to actually answer the darn questions to move to the next slide. Oh, the picture's escaping me. There was a really good picture at one time. There it is. Ha-ha! I found it. Right? So notice here, see how it's speckled? Can you all see that? Right? Where others in that same kernel of corn, right, some of them are actually completely, this is more of a purple color, and others it's kind of a red color, where you get this change that's happening. So she noticed this and she's trying to explain it. So her work, y'all, what's so amazing about it is that at the time when she was doing this and when she published her papers in the 1950s, guess when she was awarded the Nobel Prize? 1980s. 30 years after she originally did the work, because it was so groundbreaking at the time. Other scientists couldn't believe it, right? They're like, nah, this isn't happening. That's not what's really happening, right? You know, there's no way she figured this out with what she was doing, you know. And so it was pretty amazing stuff. And then, as Clint was saying, because of her work, a lot of the stuff that scientists do in biotechnology, her work and some of the others' work, is how we're able to actually know how to manipulate, say, bacteria, like that example he gave, of being able to insert genes into their chromosome and get them to produce human insulin. And what you guys probably don't know, too, is that they've actually tweaked that protein complex. It's a protein, and that's why, unfortunately, diabetics have to inject it, because if you ingested it in a pill form, what would happen? Your body would just digest it as if it was a protein, right? Where your body, your pancreas, the beta cells in your pancreas secrete that right into your bloodstream, right? And then it goes throughout the body to tell the cells, hey, sugar's here, you can eat it now, right? The problem with that protein, too, and proteins in general, is they're affected by temperature, right? Temperature can affect their overall structure, right? And that's one of the things that we didn't go over, but I definitely want you to look over. It's one of the objectives that's bolded, is the structure of a protein. So at the very basic level is all the amino acids strung together, right? But then they're going to form secondary structure where they form alpha helices and beta sheets. And again, the modeling that Paulus did for proteins is part of what Watson and Crick looked at to be able to do their ball and stick diagram for DNA structure. So then those proteins will form that secondary structure and then a tertiary globular structure as those things fold back on each other. And then some proteins, as I said, are made of multiple polypeptides. So the polypeptide is a long string of all the amino acids. And remember, all that information is encoded where? How do we know how to make all our proteins? It's in the DNA, right? It's in the DNA. So again, remember, we've cracked the code for that, and by no means do I expect you to memorize that code, but I do expect, and I want you guys to be able to use this code, right? And so this is just like when you were a kid, right, and you did like the decoder ring stuff, right? Or maybe you weren't one of those kids, but I was. So I put together, and I'll post this one for you guys too, a PowerPoint for that. I thought I had it open. Called the code. The code. Okay, so, right, here's the DNA, right? The template strand, notice that's the strand that is copied to make the messenger RNA. What do we call this other strand, which is the same code except you're just switching the T's for U's as RNA? It's the coding strand, right? So remember, 2 would be 5' to 3' if it was going in this direction. Which do you think it would go in that direction? Do you think it's going from here to here? Or do you think it's going from here to there? There's a clue here as to why this is the beginning. The start codon, AUG, which actually codes for an amino acid, right? What does your chart tell you? What amino acid does it code for? Methionine. So for the next codon, right, if we're looking on our chart, what amino acid would that be if it's CCG? Proline. Yep, proline. Right? And so we could go through and we could write the whole code for this, right? I won't make you do a code this long on the test, but I can guarantee you. Right? So bring back this sheet with you, right? Or bring this chart with you, because I want you guys to be able to do that. So let's do one together. This one's 10 long. This one's a little bit shorter. This is a sequence from your book. But would this whole thing be translated into a protein? No. Where would we start? We would start at AUG, which notice is right here, right? Before that, there are sequences that allow the ribosome to bind, right? The anyways. particular name to the guy who discovered the sequence where the ribosome binds. Can't say his name right now. Okay, so but again, if this is a bacteria, right, for the most part, this is going to be methionine, right? And then the next one, right, we would keep going until we reach what? A stop codon. Is that last one a stop codon?
Yes.
Right, that is a stop codon. So then these would not be used, right, to make the polypeptide. So this, out of this segment, right, and notice again 5' to 3', so we know we're going in that direction, right, 5 to 3. We would start out with methionine. What's going to be next? GGC. Let's see if I did mine right. Glycine, yep. Yep, I would just put the 3-letter abbreviation there. AAU. AAU. Asparagine. Asparagine. What's the next one? AAA. All A's. That's what we want, right? Lysine. I'm slow on this chart. I did it earlier. Did I get it right? Yeah, lysine. Okay, I'll let you guys do it. CCG. Is it proline? Yeah, that's what I got. GGC. Glycine again. Okay. CAG. Yeah, I think it's glutamine. Yep, glutamine. Glutamine. And then the last one says stop. So how many amino acids?
8, 7.
I counted 7. 1, 2, 3, 4, 5, 6, 7.
Oh, I thought stop also counted.
Stop doesn't count, right? Stop is stop. It's not an amino acid. Where start is an amino acid, right? Do you think a functional protein would actually be this short? No, but I promise I won't go over 7 on the one on the test. I didn't go over 7, the one on the test, right? So I have a short segment, right? You got to know, right, that stop means stop, right? And then you'll have your chart so you know where the start codon is, where you would start. And that's methionine. Okay. So Tuesday, bring your notes, right, according to this. And then Tuesday we'll give you a sheet like this for the final that will have all the objectives for the final exam, just the ones that are on the exam for you guys. Cool beans? All right. So as far as review of test 3, because we're going to do it in class on Tuesday, I will, by Tuesday, too, have my office hours for final exam week posted so that you guys know when's a good time to come by and look at all of your exams if you would like to do so, right? But I will bring them with me during class, but if you guys get done early taking your test and you want to look at your past ones, too, I will do that on Tuesday. But I suspect for 3, you're going to want to come look at it. Yeah, I posted it, too. And I'm going to post this right now, too. It is right there. And then I'm going to post the code one. I'm just going to post it right here. Desktop. Oh, I didn't upload it, so it's not going to be there. I have to go to my actual thing. So there's that just short one we just did. So make sure you bring this back with you, right? So you have the code. Or maybe I'll squeeze it on the test in that section too, so it'll be right there in front of you. We'll see. Probably should do that, huh? There's a better one out there. I think I might print one that prints better. It's not so grayed out.